Hi everyone, thank you very much, Jim. It's a pleasure to be back on stage this year, and I really enjoyed the spotlight session right now, so thank you very much. And I also enjoyed actually walking to stage to a song of one of my favorite bands, which relates a little bit to the title of my talk today, where we try to use real-time genomic approaches to better understand and contribute to the improvement of One Health. Now, the concept of One Health describes that all of the health on our planet is inextricably linked. So the health of humans, the health of all other organisms such as animals, plants, and also microorganisms, and also the health of ecosystems. Now, our last pandemic, the COVID-19 pandemic, is a very good example for the importance of the One Health concept because it has been related to the loss of wild habitats and to the loss of biodiversity. And this is something that has really been accepted by the international research community as well as by political leadership. Now we want to use real-time genomics to better understand One Health. And obviously at such a conference like here, I talk about nanopore sequencing when I talk about real-time genomics. But also nanopore sequencing technology is really the only way of conducting real-time genomics nowadays. And I'm not going to go into detail about how nanopore sequencing works for an audience like this, but very briefly, what we leverage is that a single-stranded DNA, also RNA, passes through tiny nanopores embedded in a membrane, thereby disrupting an ionic current, and this results in a disruption signal, the so-called squiggle signal that we can then, using bespoke algorithms, base call into DNA sequence. Now, the importance here is that we can actually sequence the native DNA, which means we can get very long DNA reads, basically as long as the DNA fragments are, and I will show a little bit later how we can leverage that. But we can also get information about all sorts of other modifications of this DNA strand, such as epigenetic modifications. Now, if we couple these bespoke algorithms that I talked about with powerful compute, we can really generate and analyze data in real time. But for an actual real-time application, I think it's also really important that we have portable sequencing devices that we can bring to the point of care, to the point of interest, to directly get results, for example, in the clinical setting or in the field. Now, I want to talk a bit today about how we can use real-time genomics in the context of One Health. And I guess the spotlight session right now actually provided some really cool examples already. And many of you will be working on related research here today, and I look very much forward to discussing with you in the next 2 days. I'm very happy to say that we assembled an international and interdisciplinary team of nanopore researchers and put together a perspective and review article that is currently under review, but I made the unreviewed version of it available for you via this QR code, so really would appreciate any feedback from the community. In this article, we discuss how real-time genomics can be used to improve our understanding of One Health. And this really ranges from biodiversity monitoring over generating reference genome data for wildlife, such as via the ORG1 initiative, to assessing pathogen spread within wildlife, which obviously relates to thinking about zoonotic potential in wildlife. We can assess the environmental microbiome and check how anthropogenic impacts such as habitat destructions change our environmental microbiome and therefore its functions. We can monitor wildlife trafficking, more in terms of thinking about human society and human health, which we have heard a lot about today already. We can think about pathogen monitoring in the clinical setting, detection of resistances, but also contribute to food security. We further talk, when you see here on the left side, about in this perspective article how the model-free data modality of the nanopore read data, of the squiggle data, can be used in all these sorts of applications to give us really additional information, and I will bring forward an example about that later. Important it is here that we actually include community, and I mean here by the scientific community as well as other stakeholders, when we want to conduct real-time genomics in situ at the point of care. And also, we emphasize the importance of decentralized data. Now, we obviously also mention a few of our own research examples in this perspective, ranging from applying nanopore sequencing and remote islands in New Zealand, work from my postdoc where we monitor the spread of pathogens in critically endangered wildlife, to the application for biodiversity monitoring directly in biodiverse rich areas, such as in the middle of Ecuador, so that's a project set up by the In Situ Lab Initiative, to applications on farms where our collaborators actually used it to monitor the spread of cassava virus across cassava plants and actually contribute to food security. And all of this, like the real-time genomic, the real-time application of this is obviously only possible if we also develop the laboratory and the computational parts to be able to bring nanopore sequencing into the field. And if we do it the right way, there is, I think, a huge potential to contribute to education, both at schools, universities, research institutes, and to really include stakeholders, such as, for example, the farmers I just mentioned, as well as conservationists. And here I just quickly want to relate to my plenary talk from last year that is still available online, where we used nanopore sequencing to monitor the health of the critically endangered kakapo species in New Zealand. Now, I want to talk about a few examples of my own research group that I was lucky enough to be allowed to establish roughly 1 year ago in Munich. And what we do in Munich now is like a little bit of everything. So we are still working on the biodiversity monitoring side, expanding our collaboration with New Zealand, but also with many other countries, and working directly with the IUCN Conservation Genetic Specialist Group. We further use nanopore sequencing for ecosystem monitoring, such as assessing quality of fresh water. And here we have actually started like a big collaboration with Brazilian partners who I actually met for the very first time at London Calling last year. So go out there and socialize, I guess, is the message of this. We also expand the ecosystem monitoring to assess the air microbiome or air quality, where we are using cyclonic technology to really sample and then sequence air at a high rate. We use nanopore sequencing in the clinical setting, like thinking about transmission of pathogens, both in the clinic in Germany as well as in like low and middle income countries. And finally, as I mentioned before, we can think about how we can use non-invasive sampling to monitor the spread of pathogens in wildlife, contributing to wildlife health, but also again thinking about zoonotic potential. And we also, in the end, work on the actual squiggle data, so the disruption of the ionic current data, and explore what sort of information we can get that we can directly leverage in the context of One Health. Now, I'm very happy that my entire research group was actually allowed to come to London Calling this year. They all have posters, so if you're interested in any topic, please find them. Plus, Harika is going to give a lightning talk later today. So, but let's jump into like some examples. So the one question we are asking is how can we use non-invasive sampling, such as of water, air, and soil samples, to better understand the ecosystem health and consequences for human health if we disturb the function of this ecosystem health. So for this, we are basically assessing the entire genomic material in any of these non-invasive samples. In the very first study that I actually conducted with nanopore sequencing, which I still did next to my PhD in Cambridge, we established nanopore sequencing for freshwater monitoring and actually used our local river Cam in Cambridge as an example and sampled water at different time points, like really along a gradient of anthropogenic impact, so ranging from upstream of the city, like a quite natural habitat, throughout the city with like really high pollutions at what you see here, the location 7, for example, a huge anthropogenic impact, to the outlet of a sewage pipe from a close-by wastewater treatment plant. And we back then used long-read amplicon sequencing via nanopore technology to assess the bacterial diversity of these water samples. And here I just show you like one of the results of our study back then, where we are looking into bacterial genera that are potential human pathogens. So these are the first rows of bacteria here, and also into bacteria that have been associated with wastewater treatment. Now, the important part is on the right side here, where you again at the bottom see like all the sampling sites, so basically along this gradient of anthropogenic impact. And the color coding shows you how enriched like certain bacterial genera are in any of these sites. And I hope you can appreciate that at like the sample 7 that I just pointed out at this like hugely anthropogenically impacted location, we find like an enrichment of potential human pathogens. Whereas at the site 8, which was really the output of this sewage pipe, we find an enrichment of wastewater-associated bacteria, which makes a lot of sense that we are finding the signature there. What worried us a little bit is that we also found an enrichment of some potentially very virulent pathogens downstream of this wastewater sewage pipe outlet, which made us hypothesize that the general microbial community was changed by anthropogenic impact like this, like changing the ecosystem in such a manner that potential pathogens can flourish, like a prime example of One Health. Unfortunately, with this sort of approach, we were not really, with high confidence, able to go down to the bacterial species level or to do any functional sort of annotation. So we decided to follow up on this research and actually do shotgun sequencing with nanopore, so really sequence all the genomic material in the samples. And this allows us to do de novo genome assemblies from this data, or create so-called MEGs or metagenome assembled genomes that give us really high, like, confident availability predictions or presence prediction of certain bacterial taxa, also of all sort of other taxa, such as eukaryotes, fungi, and so on. Plus, we can do a functional annotation. We can look for biosynthetic gene clusters that tell us something about the function of these organisms. And we can also like use the shotgun sequencing data and just align it to databases to get like a quantitative idea, or to do like reference guided assemblies. So we went back to this sewage pipe outlet that we were interested in Cambridge and processed the water both by shotgun sequencing as well as by 3 replicates of the amplification-based approach that I just showed you before. And here, just like on the taxonomic phylum level, you can already see like huge differences between the assembly-based approach and the other approaches in terms of quantification of the taxa. But that obviously makes a lot of sense, because for the assembly-based approach, we only really find the high confidence, so probably highly abundant taxa. At the same time, we see that the shotgun alignment-based approach in terms of quantification actually correlates very well with the amplification-based approaches. And so this like really showed us, and please refer to Anastasia's poster for more details here, that shotgun sequencing approaches have like a lot of promise, because we can get a rough quantification of microbial taxa using the alignment-based approach, while the assembly-based approach can give us like really an idea of the functional annotation of potential pathogens as well. Now, this motivated us to go on and analyze the air microbiome or the air ecosystem in the very same manner. Because while we know a lot about the physical contamination of air and how it has an impact on human health, it's actually one of the biggest environmental threats for human health. We don't know so much about the biological contamination, and bioaerosols have actually been really understudied. So we started a collaboration with a global health institute in Barcelona, and now try to like assess how these anthropogenic impacts change the air microbiome with potential, again, human health repercussions. And in a very small pilot study that I want to show you right now, we actually took replicates of air samples and water and soil samples at the very same location and processed those samples in the same manner using shotgun nanopore sequencing. And I hope what you can appreciate is that we actually find like very specific water and soil microbiome, and that the air microbiome obviously differs, but also seems like relatively characteristic on its own. And interestingly, we find that the microbial diversity in these air samples, both in terms of the actual richness but also evenness, is higher than in the other samples, really motivating us to go further and study this in more detail. So what Tim in the lab did, he would go into like a more controlled environment, into a greenhouse on our campus, and sample air, again using the cyclonic technology. And here you see like 3 air samples from like 1 hour of sampling that were taken like consecutively, one after the other. And we see a quite high variability between those samples, which is not a good thing, right? Because we can therefore not like assess the microbiome in a robust manner. We did this again on another day, again a lot of variability, and the third time we see the same thing. But then we actually went in and sampled for 2 hours in a row. And you see that actually after 2 hours of sampling, using just nanopore shotgun sequencing, we can assess a quite stable air microbiome. And especially after 3 hours, like we see a really robust signal across different days. So obviously this is a controlled environment, but it shows us that if we have a stable air microbiome, we can actually assess it just using shotgun nanopore sequencing. And I importantly have to say that we use the latest R10 chemistry for this, with the increased sequencing accuracy, but also the rapid barcoding kit actually really allows you to put tiny amounts of DNA in, which was really promising. And for the 2 hour and 3 hour samples, we are even able to build assemblies now, so do functional annotation of the air microbiome based on long-read data. Now, if we come back to this overview of where we are thinking of how we can use metagenomics to assess like ecosystem functions and ecosystem health, one problem is if we only rely on DNA, that we don't necessarily know if the DNA comes from living or dead microorganisms. But this is obviously important because only living organisms actually contribute to ecosystem function, and it's especially important if you think about pathogen monitoring, because we're obviously only interested in pathogens that are still pathogenic and virulent. And one important application like in pathogen monitoring is obviously in the clinical setting. So just really briefly an example where we, for example, Ella in our lab is monitoring bloodstream infections that are caused by gram-negative bacteria that are actually in theory, relatively easy to cure using like antimicrobial therapy, but the problem is that the resistance profiling that is currently used in the clinic can take like many days before like giving any results back. So that really means that normally these sorts of approaches don't inform any therapy in the clinic. Whereas, as we all know, we can get nanopore sequencing results, including the sample treatment, within 2 or 3 hours. And this is just the result of one blood sample that Ella analyzed, where we find on the long reads, like really evidence of biosynthetic gene clusters that contribute to resistances. And when we then compare these predictions with the actual experimental results that we would get many days later from the clinical data, we see that we can indeed confirm some of the resistance predictions. And in the case of the ciprofloxacin, which is actually one of the fluoroquinolones, we see susceptibility, but obviously we could only predict a reduced susceptibility here. And I know that many others, and we as well, are working on like actually making these approaches also on the computational side more robust to take these sorts of uncertainties into account. But coming back to the question that I had, right? So, like when we find pathogens or any other organisms, do we know if they are, if this DNA came from dead or alive organisms? So we had a little bit crazy idea that maybe we could leverage that squiggle data detects differences on the atomic level in the DNA, and use this to distinguish dead from living DNA or DNA from dead and living organisms, under the assumption that when a microorganism dies, the DNA gets exposed to environmental damage, such as increased temperature, humidity, UV. There are no repair mechanisms happening. There are lots of enzymes around that are degrading the DNA. And our question was, does this leave like a characteristic signature on the DNA that leads to a slightly different squiggle data? And there, I suddenly realized, wait, I have my own research group now, so I can actually follow up on some crazy ideas. So we started like just growing some E. coli and let them die, and then nanopore sequenced both the living and the dead bacteria. We then fed this data into a residual neural network, which in this sort of way has been used like in many other like read-based classifications of squiggle data, and it importantly consists of like all of these bottleneck structures that helps us with controlling like for any sort of degradation while training the system. And importantly, we trained this model like using our data to then give a binary prediction of alive versus dead. And I will show you some very preliminary results of this, but please check out the poster of my team members, and also Harika will talk a bit more about this later on. But we get some promising results in that we get a relatively good sensitivity of this first model of 0.77, which actually means the model is able to detect living bacteria relatively well. And obviously all these like performance metrics can be improved, so we can work a lot on like model choice and hyperparameter optimization. But the one thing that really worried us was the specificity, right? Because it's like basically random if the model classifies a dead DNA as dead DNA. But we by now think that this might have been a problem with our training data, where we probably have a mixture of dead DNA, so DNA of organisms that died on different days. So we are also currently creating more data for this. But I think the really important point is that if we can manage to get like a model like this together, all of you could just go back and like apply it to your squiggle data, and we would get sort of an idea of if this viability thing is a problem at all in our metagenomic studies, or if all microorganisms were alive anyways. And now I really briefly, to wrap up, want to come back to our perspective article. Because there we also like a little bit more broadly discuss how real-time genomics really has the potential to contribute to more equity and inclusion in One Health research because of its distributed and also disruptive nature. And obviously this has to do with accessibility, and we know that nanopore sequencing has been developed to be available to everyone, everywhere. This relates to costs, so that we have like basically absence of any sort of like upfront investment costs or very low upfront investment costs. Here, however, we have to say that the price for the consumables is obviously still significant, and this is still a problem for accessibility to low and middle income countries, who, like, in addition, suffer from often like a lack of availability of distributors. So that in the end, the shipping and the material costs are even more expensive for them, or sometimes they don't have access at all to nanopore technology. And there we obviously have to change a lot to really create like an equitable way of taking up real-time genomics. The accessibility also relates to automatization, obviously, so we want to give this technology also in the hand of non-experts, so automatization both on the laboratory and the computational sides, where I also know lots of you are doing research on. And this would really enable us to include the community more, so the broader scientific community, but also all sorts of other stakeholders, right, like farmers, medical doctors, and conservationists. And I think this has this promise that we can actually do something about what is still ongoing problem, and what we are calling the neo-colonialism or helicopter research, right? That like rich, Western institutes and researchers actually do any sort of research without consultation. And in the long term, I hope that this can like both diversify and democratize the genomic research, and provide self-determination to people all around the world, especially to indigenous people. And here I'm super happy that we have some Maori authors on this perspective who are talking about the concept of ahika, or keeping the home fires burning, with which they mean that indigenous communities often require that data and analysis and so on to some extent stay within their communities. And I think there's a lot of like promise here. Obviously, if we think about this lab in a suitcase idea, which is exciting on its own, but if this is done without community engagement and inclusion, this can also have like really risky repercussions, in that we are actually promoting helicopter research in the end. So we have to be really careful there as well. But I hope that I showed you that obviously to really achieve equitable inclusive access to real-time genomics in the context of One Health, there's still a lot to be done. like global inequities have to be fought, which is a completely different project. But I think we can also do something on the technological level and think about how we can make the technology more flexible with respect to, like cultural considerations, also commercialization and infrastructure especially. But I think then it really holds the promise that we can on-site create a lot of data that we can then leverage to really better understand repercussions or implications of One Health that have so far really remained hidden to us. So I thank you very much for your attention.